Welcome back once again to our lectures on fluorescence spectroscopy. This is going to be part two of our lectures on protein fluorescence, meaning intrinsic protein fluorescence. And today we're going to stress the time-resolved measurements of protein fluorescence. There might be a part three. I'm thinking about some unusual phenomena that I couldn't fit into these first two lectures, but we'll see about that later. So, as I indicated previously, proteins can have really almost a infinite number of different conformations with the number of amino acids that they have to use, the 23 amino acids. But we're only going to present some general features of how protein structure is linked with the time-resolved decays of proteins. It's not meant to be an exhaustive list, just some comments to give you a feel of what factors are important. So first, let's look at, since we're going to look at time-resolved measurements, let's look at the intensity decays of tryptophan, which is the dominant emitter in proteins. And this has been controversial for decades, and I'm really thankful that it's finally solved. But what was found early on was that tryptophan had two decay times, a longer one, 3.3 nanoseconds, and a shorter one, around a half a nanosecond. And this really bothered a lot of people. The sitting on grant review panels, people would complain that, well, how can you look at proteins when you don't understand tryptophan? I'll get to the final conclusion in a minute. But after all of these studies by many people choosing different Hamilton, done different instruments, different Hamilton emission filters, the final conclusion was yes, tryptophan in neutral solution does have two decay times. Shown here is 3.4 and 0.5 nanoseconds, but the intensity of the short lifetime component contributing to the total intensity is less than 5%. So it's a very small number, and it really didn't matter that tryptophan in neutral pH had two decay times. The reason it didn't matter is because once you put tryptophan into a peptide chain, well, now you have derivatized the amino group and the carboxyl group, and when you do that, the short lifetime component disappears. So we call this model compound NADA N-acetyl-tryptophanamide. And if you look at the intensity decay of NADA, it's in water, it's basically a single exponential, or if you do the frequency domain measurements, it's also clearly a single exponential. So I always thought it was unnecessary to be worried about tryptophan, but many people's were, and I think this observation has sort of cleared things up. So we don't worry about its non-exponential decay in water at this time. What's the origin of the short lifetime component? Well, here let's look at the lifetime and the fluorescence intensity as a function of pH, and they track each other perfectly, which says whatever is going on is a dynamic process. It's happening during the lifetime of the excited state. And so what causes this? Well, there's a rise in intensity right around pH 8 or so. This is just the region where you are deprotonating the amino group on tryptophan, and when you deprotonate that amino group, it's no longer able to donate a proton, and as you know, indole is an electron deficient species, So it would like to pick up that proton. So you can draw any number of conformations. These line diagrams might not be informative, but they might suggest that one of these conformers is the origin of the other decay time. But I don't think that's been proven absolutely. But it's one way of thinking about it. So the short lifetime component in tryptophan is due to an electron transfer, or I guess a proton transfer to electron deficient species. Now let's go to tyrosine, which is the other aromatic amino acids that we speak about. And of course, we can have tyrosine or N-acetyl tyrosinamide, which is a derivatized version. If you look at the derivatized, if you look at tyrosine itself, in buffer, it's purely a single exponential, so we don't have this complication we had with tryptophan. This is something I don't understand and hasn't been paid attention to that I've seen in the literature, but the N-acetyl tyrosinamide in this case has a shorter lifetime component, which is not understood. The one thing that can make tyrosine multi-exponential is the loss of a proton in the excited state. We discussed how the pKa of the hydroxyl group drops down to a very low number, pKa 2.5 or so. So if you're a neutral solution, the moment the molecule absorbs light, starts to lose a proton. When it starts to lose a proton, you start to get what's called tyrosinate emission, which has a shorter lifetime, and you can prove that it's due to the hydroxyl group. Of course, if you do the experiment with O-methyl tyrosine, where it's not doesn't have any proton to lose, so it's a single exponential. So tyrosine also has its components, but the tyrosine, I have trouble saying tyrosinate emission is very minor compared to the total tyrosine emission. So how are the spectral properties of proteins linked to their three-dimensional structure or their being in a native state in solution? So I chose one of the best examples of this is ribonuclease T1. It's a single chain nuclease, relatively small. The protein, the tryptophan, is mostly shielded from water, but it's not shielded like it was in azurin, where there was no water contact at all. But it naturally is sensitive to being in the folded state or the unfolded state. So here, if you look at the emission spectra here, the excitation wavelength must have been 295 nanometers, because we didn't filter it out very well. So this has nothing to do with the T1 emission. But you have a blue shifted emission in the native state, typical. And if you take it all the way to the denatured state, you shift all the way out from 320 to 350. So there's no doubt you can say something about the protein conformation and how it folds or unfolds by its shifting of the tryptophan emission. This next slide will take a little more explanation, but we are supposed to be talking about time-resolved fluorescence. So we went over this part of the spectral shift, but what happens to the decay times when we go from the native state to the denatured state? And looking at the frequency domain data, we have the frequency response for the native protein, which has the nice symmetrical single exponential response. And in the denatured protein, the decay becomes much different from the single exponential shape. It's obviously a multi-exponential shape. Sorry I didn't put down the numbers here for you, but it's at least a double exponential, maybe even a triple. The other thing that happens, which I think is more important, is that whenever you denature the proteins, you free up their ability to rotate. So these are the differential phase measurements we spoke about early on, where we're measuring the phase angle between the light that is emitted parallel to the excitation light and the light emitting perpendicular. For a isotropic rotator rotating freely in solution, you have this Gaussian-like structure which conforms to the insert here of a single correlation time decay. When you denature the protein, the anisotropy decay becomes more rapid, and it also becomes multi-exponential, as you can see in the insert of this slide. In the frequency domain, these rapid motions of the tryptophanes always show up at higher phase angles and higher frequencies. So for those of us who have looked at a lot of these frequency responses, we knew immediately this meant, there's a sub-nanosecond correlation time in there, probably 100 picoseconds or so. So the modulated anisotropy says something, same thing. Complicated term, I didn't talk much about it in the previous lectures, but for the moment we can consider it to be like a steady state anisotropy, especially at low frequency. So the steady state anisotropy at low frequency is high, and when we denature the protein, the steady state anisotropy goes down because the tryptophanes are rotating freely. So this is a great example of linking the state of the protein with the intensity and the anisotropy decays. This was only possible because of this very simple decay we had to start with for a single tryptophane, and all the other ones we're going to see are more complicated than this. So the anisotropy decays are very different among single tryptophan proteins, and more importantly, the anisotropy decays are much more variable than the intensity decays. So I've put here 3 different proteins for you with their anisotropy decays. So the first is liver alcohol dehydrogenase, which has 2 types of tryptophanes in it, but the proteins always show a rapid decay before they show the part of the decay due to overall global rotation of the protein. So for this protein, there's a short correlation time around half of a nanosecond. Some proteins that have very exposed tryptophanes, the anisotropy decay is even more rapid at the beginning, and in fact you can't even see the global rotation here because all of the anisotropy is lost in the first 200 picoseconds, or we get to phospholipase A2, a particular tryptophan in there, and it has even a faster initial anisotropy decay. And the reason I'm bringing this up is because this is a characteristic feature of the anisotropy decays of almost all proteins, regardless of the molecular weight. So most proteins show some flexing of the tryptophan residue before the protein settles down and just undergoes global rotational diffusion. I put together a case where I thought we could imagine these two situations. So I don't know why I chose the retinol binding protein. It has a number of tryptophanes in it. Some are on the surface, some are buried in the interior, and it's pretty easy to imagine what the anisotropy decay of this protein would look like, because you know there's going to be some rapid part of the decay, and then there's going to be a slower anisotropy decay due to overall rotational diffusion. And this was a very interesting plot, and I cannot remember the author, but I sort of decided at the beginning I shouldn't be using author names because there'd be too many of them. But almost all proteins display this property: a short sub-nanosecond correlation time, and then a long overall rotational correlation time. So here we have those two numbers listed for the segmental motion as a function of molecular weight, and the global motions as a function of molecular weight. Of course, the global correlation times become much larger, but pretty much you get the same short segmental motions of the tryptophanes independent of the protein molecular weight. I'm giving you this example for two reasons. One is proteins like hemoglobin have been looked at for years, trying to find some tryptophan from emission. In this particular protein, we have one tryptophan in it and a heme group, and the heme group is a very effective acceptor by resonance energy transfer, and some 97% of the energy goes into the heme group, so the tryptophan is only weakly fluorescent. If you look at the intensity decay of this protein, which is a peroxidase, you see a very rapid initial intensity decay, and then a much longer, slower intensity decay. How do you interpret this? And this sort of goes along with my feeling you should never get too preoccupied with small components. But let's look at the lifetime distribution. This is the analysis where we plotted the amplitude associated with the correlation time versus the correlation time. And in terms of area, we see 97% of the anisotropy decay area is associated with this very short lifetime. And then we see there's two other areas that have short lifetimes. So what do you do with this? My initial response would be it's probably an impurity, ignore it. Another possibility, it's due to the kappa squared effect that we discussed for energy transfer, where transfer can be affected by the orientation of the transition moments between the tryptophan and the heme. But in my opinion, these are just short correlation times, short amplitudes in the decay time that probably have nothing to do with the protein itself, but are rather impurities. So now I chose another protein that has a very short component in the intensity decay. I think I showed you this protein once before, but this is part of a protein from a peptidyl prolyl isomerase. Some of you may remember from your biochemistry that prolyl is a restrained amino acid, and when it forms a peptide bond, it can be in a cis or trans formation, whether it's in a cis or a transformation, is not determined when the protein is being put together. So originally you get a racemic mixture, and the cells need a protein that will isomerize this as needed so the protein can fold properly, because if it's not in the proper cis or trans position, the protein won't fold properly. So again, we used site-directed mutagenesis. We're looking at the protein with one tryptophan missing, or the protein that has both tryptophans in it. When both tryptophans are in it, you get the normal short decay, normal intensity decay, and this is the lifetime distribution here for the protein with two tryptophans. So it's sort of the usual set of lifetimes of a sub-nanosecond one, a nanosecond one, and a dominant 6 nanosecond ones, which we'll see is sort of a common feature of many proteins. And that's this decay up here. When we got rid of the longer-lived protein, we wound up with a much shorter intensity decay. The lifetime distribution is shown down here, and here most of the lifetime is very short. So it's not only a heme group, it's not only a energy transfer acceptor that can cause a short decay time. So there are certain features in protein structures that can quench the tryptophans, and I don't know if the mechanism of this has been worked out, but I was very surprised to see that a nearby phenylalanine, which is nonpolar, is able to quench the tryptophan. That just didn't seem reasonable to me, because the phenylalanine is nonpolar and the tryptophan's polarity sensitive, but this is certainly the way it is. So short lifetime components can appear even if you have no other absorbing groups in the protein. In many cases, probably most cases, it's really impossible to identify which of the tryptophans are associated with which lifetime in a multi-exponential lifetime. So taking a most extreme case, I have this monomer here. It's a beta-galactosidase. This is only the monomer. The monomer has 17 tryptophans in it, so you're never going to resolve those. You heat the protein up, all of the frequency responses shift to higher frequencies, meaning the lifetime is getting shorter. You can do a lifetime distribution analysis for this, and you get two major components at 5 degrees and two major components at 85 degrees. I really don't, I'm sure if you did a 3 lifetime distribution analysis, you'd probably wind up with 3 components. Even though these lifetimes look like they represent 2 different classes of tryptophans, I wouldn't agree with that. I would say we're not sure what these 2 lifetimes look like. They're true lifetimes. They're not apparent, but they're apparent distributions. You can't really say they represent something physical in the protein without a lot more experimentation. If you do a lot of work in some proteins, you can understand where the tryptophan emission comes from. So this is a TRP repressor, and as we indicated in the previous lecture, the TRP repressor turns off all of the genes associated with making tryptophan because it's energetically expensive to make tryptophan. So the researcher here did enough experiments so he could separate the 2 emission spectra from the wild type protein. So in the wild type protein, which is a dimer, there's two sort of exposed tryptophans, the W99 here at longer wavelengths, and buried tryptophans, the W19. Let's see. Well, I got that. This was the wild type. This is at the shorter emission wavelength. And excuse me if I'm not quite reading them correctly, but I'll just come back to something in a second. You can see that they had different lifetimes because the frequency response is different for the one tryptophan as compared to the other tryptophan. And as might be expected, the wild type frequency response or the wildlife mean intensity decays in between them. But one of the reasons I chose this example is even when we knew all of the stuff about the trip repressor as to where the tryptophans are, how to separate their emission spectra, it's really still not understood why the total emission spectra is greater than the sum of the two individual emission spectra. So that's a mystery. Certainly, it has to do with how the protein folds when the tryptophans are substituted. But in any event, they don't add up, and that's sort of something that happens a lot in protein fluorescence. We can't always make things add up. Here is something that has bugged me for a long time, and I got myself in a lot of trouble for talking about this. But first I'll show, let's look at some triple lifetime intensity decays, triple exponential analysis. I've always been sort of critical about them, but I'll tell you about why for a moment. So let's look at first at the BSA, the reports from different laboratories. You'll always notice for this single tryptophan, human serum albumin protein, there are always a short lifetime, a short lifetime here, a fraction of a nanosecond, another one on an average of 2 nanoseconds, and another one on an average of 6 nanoseconds. If you look at a number of multi-tryptophan proteins, you sort of find the same answer: a short lifetime, an intermediate lifetime, and a long lifetime. So this always bugged me, and I always asked myself the question: are these real lifetime parameters that represent something in the protein structure, or are these numbers that just reflect the resolution capabilities of the instrument that you're using? The number of tryptophans are listed here. So maybe you just always get these values because of your limited time resolution in the photon counting method or the frequency domain method. This brings me back to the slide that I showed you, I think it was in part 3, where we showed you the intensity decay of 3 individual fluorophores, which were each a single exponential, and then we showed you the intensity decay of those 3 mixed together, and we did the analysis and looked at the chi-squared values, and we decided, yes, of course, you can tell that a 1 exponential fit doesn't work, but could you really tell from the chi-squares that the 3 exponential fit is much better than the 2 exponential fit? And it seems to me like these 3 lifetimes have about the same difference in decay times as we saw for all of the protein fluorophores. So that made me wonder how real all of the three component correlation times for proteins are. Now, so many authors have published triple uh, exponential analysis results for proteins, and so have I. So I can't blame anyone else. Uh, but is another caveat which hasn't been spoken about very much, and the caveat is, what is there spectral relaxation in proteins after excitation? Remember when we were talking about solvent effects, and then we went on to the topic of spectral relaxation, where the fluorophore would go to the excited state, and over a period of time, then the solvent would reorient around the excited state, and it would relax slowly to a longer wavelength emitting state. So there really was no single spectrum that you could associate with that. The hallmark of an excited state reaction is a increase in the intensity at long wavelengths before the intensity decreases. So we see for this indole and glycerol, pretty viscous solution, we see initial fast decay time on the short wavelength side, 310 nanometers. On the long wavelength side, we do see a rise time before decay, and that meant it took time from the initially excited fluorophore to get over to the longer wavelengths to emitting at 390 nanometers. This also occurs, you can look at this in different ways. Let's see, two ways we described it. One were the time resolved emission spectra. They're sort of easier to understand. What does the emission look like at any point in nanosecond time after excitation? And for the system that is relaxing, you see the spectrum shifting to longer wavelengths at longer times. Another way of describing this system is to look at the decay associated spectra. So here an analysis is done where we get all of the pre-exponential factors and the tals. Not to get too deeply in the weeds, normally the lifetimes are held as global parameters, but what you get out of this is an emission spectrum which is associated with each of these nanosecond components in the multi-exponential decay. And this is the total decay here, but let's look at these other ones. On the short wavelength side, you see there's initial increase and then a decrease. It has a negative pre-exponential. You go to a half, my eyes are not that good, yeah, about a nanosecond, same thing, positive and then a negative pre-exponential, which we said is a hallmark of an excited state reaction. And then you get out to longer times and you don't see that any longer. So there's no doubt that indole and viscous solutions can undergo spectral relaxation, and a viscous solution to me seems like the interior of a protein. So I feel like spectral relaxation can be contributing to all the triple exponential decays. Another one I have here is for myelin basic proteins. That one was just indole. So here we have for the single tryptophan protein, myelin basic protein, forms sheets of protein around your nerve fibers. We can also make the decay associated spectra, and we see that the shorter decay times are always the shorter decay-associated spectra, which sort of makes sense because when at shorter times, it's the bluer emission that's appearing. And in the same way, you can look at the time-resolved emission spectra, and they continue to shift with increasing time. And to me, this complicates the meaning of the three decay times. Yes, they might be approximately correct. I'm not saying that spectral relaxation makes a really dominant contribution to the protein intensity decays, but it probably makes some contribution. And over here was just another case of a lipophilic indole derivative placed in a mice cell. But we don't need to go over that. I think I made my point pretty clear. So what do we know about proteins from tryptophan fluorescence? Well, we know some trips can be shielded from water or exposed to water. We know that they could be shifted by substrate binding. I think I showed that in one of the initial talks. We showed that the wavelengths, just about all emission spectra, wind up having the same long wavelength emission when they're completely denatured. When everything is known about the protein, you can sometimes correlate the spectral data with the structure of the protein, but you cannot always do this. Many times you don't know the structure well enough to correlate it with the optical properties. This is an important one, which we'll come to again in a minute. Minute changes in the structure can shift the emission spectra. We showed in the previous lecture how contact with just one alcohol group, serine in particular, could totally shift the emission spectrum of the tryptophan in an azurin protein. Almost all proteins display multi-exponential decays. They all show loss of anisotropies. Not everyone agrees with me with this, but I think even in globular proteins, there's flexibility in the globular regions, which is why we saw that non-polar quenchers could quench all the tryptophans in globular proteins. I guess the bad news here I put in red is that we cannot determine the overall 3D structure of the protein, cannot be revealed by fluorescence. This I consider to be a bad news slide prepared by one of my friends, where he collected data for a large number of proteins, and he looked at the emission maxima compared to the quantum yield, or the emission maximum compared to the mean lifetime, or compared to the natural lifetime of the fluorophore, the one without any quenching. And basically, these plots say that there's really no known correlation between the protein structures and the spectral parameters, unless you have done a detailed study and you know something about the proteins. Now this is interesting. Let's see, I've been teaching biochemistry in the medical school for 30 or 35 years, and whenever I taught protein structures, I would tell the students, we have the genetic code, we can know the sequence of many proteins, but even if we know the sequence of the proteins, we do not know how that sequence will fold up in what type of structure. Stated more simply, knowing the sequence of the protein did not allow you to predict the three-dimensional structure of the protein. Artificial intelligence is changing this. So now there's a number of AI programs called AlphaFold, AlphaFold 2, a number of names, but given the genes for the protein, or the genetic sequence, they can then get the amino acid sequence, and they can predict the three-dimensional structure of the protein. And this seems to work pretty well. It doesn't work for all cases, but for many proteins, it's done a very good job of predicting the 3D structure that compared very closely with the X-ray structure. The programs do have some problems, just in case you're an AI type of person who loves this. I find that the literature pretty intimidating to try to look at, but the programs still have trouble with protein-protein bindings. They haven't been good at predicting that, or protein assemblies. This was a real disappointing one. It was thought when you could predict the 3D structure of proteins, well, you could predict the structure, the antigen binding site of an immunoglobulin, and then you could see if that antigen binding site matched the drug you were trying to develop, and it found out that that didn't work very well. You couldn't predict the antigen-antibody interactions from using the present AI programs. These problems, I think, will be solved in the pretty near future. But there's one problem that I don't know how this is going to be solved. And this is the problem of how do you link fluorescence with the AlphaFold technology. And if you consider the size and distance considerations, they're very different for the programs. For fluorescence, the things that are affecting the emission spectra are very small, close-range interactions, polarity around the ring, hydrogen bonding, effects of other aromatic amino acids. This is a level of detail that I don't think is yet included in the AI programs. All the AI programs for protein folding are made by looking at many, many protein structures that have been determined by X-ray and letting the AI figure out how this predicts how you can use this to predict the protein structure. So you have to have a training set of data for the AI program. I haven't seen any explanation yet where the researchers have extracted from the AI programs what they consider most important in predicting protein folding. But I think one thing that's pretty certain is that the optical properties of tryptophan have probably not yet been included into the training sets for the AI programs. And I'm not aware at this time of any predictions of protein fluorescence from the AI programs that have been published so far. So that sounds pretty pessimistic, but in time, in your generation, these things will come. So that's the end of part G. That should have said part 12B, and I'll probably see you again for part 3 of protein structure. Thank you for listening. Bye.